and welcome to all those on Facebook. Thanks for logging in, and let's jump in. Let's talk about symbiosis and slime. So what I want to talk to you guys about tonight are some of the things that I like to think about all the time. And really, I think about slime and mucus a lot. So I'm a new professor at Eckerd College. It's my second year at Eckerd. And so the stories I'm going to tell you about today really started when I was a graduate student in the University of Florida, working with Matt Szczypulski, doing some coral reef microbiology. Then I got into kind of the realm of symbiosis a little bit more than the microbiology, and went out to Stanford for a postdoc and worked with John Pringle, where we started using Aiptasia, or a small sea anemone, as a model system that we'll talk about. And then I joined the faculty at Eckerd College last fall. So the first thing, right, so I'm a coral biologist, microbiologist, physiologist. I think a lot about host-microbe interactions. And so first question we need to talk about, right, is why do we want to study corals? Well, when you see a picture like this, or if anybody's been out snorkeling or diving on a reef, right, that's a pretty easy question to answer. Why do you want to study corals? Because you get to be in an environment like this, right? So you see all these amazing colors, shapes, morphologies, different types of coral, fish, invertebrates, right? It's this gigantic rainforest of the ocean, incredible biodiversity, and also provides so much habitat and structure for not only all those other organisms, but also a lot of ecosystem services for us, right? So this is the basis of the ecosystem in the food web in tropical waters. And really, so much of our coastal protection comes from coral reefs. And so I got into this as an undergrad the first time I started diving, right? Saw these and said, okay, I don't want to be pre-med anymore. I'm going to go play on the reefs and study the corals. And so first we have to kind of dissect what really makes this environment up, right? So what makes a coral reef? And so at the heart of it, we have to talk about the corals, which are animals, right? So many of my friends think corals are just rocks, right? They are animals, they are plants, they are microbes, they're meta-organisms, just like we are, right? We're more bacterial cells than we are human cells. And so the same thing kind of goes for the coral. Driven by the interaction of the coral host and the algae that live inside of them. So these are unicellular dinoflagellate algae that are photosynthetic, provide a lot of the nutrients and energy requirements for the coral in otherwise very nutrient-poor waters, right? So there's not a lot of food floating around in the coral reef environment. And so these photosynthesizers that are living inside of the coral tissue provide all of that primary productivity and that base energy that fuels the rest of the ecosystem. And so what you see here, these are just some juvenile coral polyps. And I hope you can appreciate the corals themselves don't have a lot of color. They're not really pigmented, and they're almost translucent. The colors that we see, those vibrant colors that come from the reef, that's coming from the algae that are living inside of the tissues. Okay, so the photosynthetic pigments in those algae are giving the coral the color that we see. And so where are the Symbiodinium? So these are the Symbiodinium are these algae, these photosynthesizers that are living inside of the tissue. And corals are pretty simplistic, okay? So they're like jellyfish. They're in the phylum Cnidarians. And sea anemones and corals are in the class Anthozoa. And these are relatively simplistic organisms with only 2 tissue layers. Okay, we have 3 tissue layers, Cnidarians only have 2. And that's the epiderm, that outer tissue layer, similar to our skin, and then a gastroderm, which is part of the feeding cells of the organism. And the algae, these dinoflagellates in the genus Symbiodinium, reside within the gastrodermal cells of the coral. Okay? And they actually get there through the process as if the coral was going to start to eat them. But through that process, the coral has a trigger and can sense that that algal cell is actually a symbiont, and it's going to provide some benefit to that coral. So instead of continuing to digest it and use it for food, it encapsulates it in a membrane-bound vesicle, and that vesicle stays within that gastrodermal cell. And those algae can then start to divide and reproduce within that cell. And that's where they live their life. Okay? And as they're photosynthesizing, they're releasing that carbon, that sugar, that the coral then uses for its own metabolism and energy needs. The photosynthate, that sugar that's produced by the Symbiodinium, is in excess to what the coral actually needs. And so the coral secretes this extra sugar as the slime. Okay, so this is the slime that we're going to talk about. And it coats the entire outer surface of the coral. And it provides a lot of functions that I'll get to in a few slides. One of those functions is a very rich organic food source for microbes and other bacteria. Okay, and that's going to play a really important role later. So first, let's go, how do we get from an individual tiny coral to a really big reef, right? So what's the kind of cycle of formation of a coral reef? Well, so we start out with the adult colony, okay? Corals, most corals, reproduce by a process called broadcast spawning, okay? They rely on external fertilization. And so what happens is they release sperm and egg into the water column, hopefully at the same time, so that those sperm cells can find those eggs. Because you can imagine if two corals release sperm and egg on different days, even within a different hour, the ocean mixing is going to swirl all those gametes around, and the chances that that sperm cell is going to ever meet that egg is really, really low. So the way that they do this, and we're still kind of figuring out some of how corals know when is the time to spawn and release those sperm and egg. But they've figured it out. It's based on tides and lunar cycle. But they know, all the species within a reef know that tonight's the night and we're going to release sperm and egg. Those sperm and egg mix within the water column and they form swimming larvae. Okay? So you get these little zygotes forming, and then you get what's called a planula larvae, or larva. And these larvae can swim around through the water column, spreading out from their parental colonies to find a new place on the reef to settle out and start forming an adult colony. Okay? So you get a settlement of a single planula larva, and it starts to form a polyp. From this single polyp that's settled out, it divides asexually, clonally, to give rise to an entire adult colony that we see. So when you look at a colony of coral, there's hundreds, if not thousands, of individual polyps that are all connected together, that all originated from a single polyp. Okay? Now, as a coral biologist, how do we study spawning, and how do we go out and collect these larvae? Right? Well, first we have to figure out when the corals are actually going to spawn. And so we've narrowed it down for some of the species that I work with to maybe a 4 or 5 day window. Usually they go after the full moon in August, and these are Caribbean species that I'm talking about. And so we go down to the reef, and every night we wait. And we wait, and we wait. We always have divers in the water, and then suddenly somebody comes back up. They're going, they're doing it. And so then we have to go and collect all this sperm and egg so we can bring them back to the lab, cross-fertilize them, and start making our juvenile corals. So what does this process kind of look like? And so I've got this video that I'm going to turn the sound on. And so I said we wait a long time for this to happen sometimes, right? And so you're on the boat, and we're waiting, and sometimes we have to get the corals into the mood, right? So there's hours of berry white playing on the boat. And so I want to set this up a little bit. So this is a colony of Orbicella favulata. So this is one of our bolder species in the Caribbean. And so this is just a diver with his light waiting, right? And so we wait, and you can kind of see what happens at night with these spawning events is all the other species on the reef know what's going to happen too. And so all the brittle stars come out, all the little fish come out, because sperm and egg, lots of calories, lots of nutrients. So they're all ready for a meal. The corals are ready to get it on. And you get an entire release of these sperm and egg bundles across the entire colony. And if we panned out, we'd see this happening on coral colonies all around. Hundreds of thousands of these little gamete balls. So these are sperm and egg together that then break apart. The sperm go find other egg cells, the egg find the sperm, and they float to the surface because they're more buoyant than the seawater. And then we rely on external fertilization. And so you're just swimming through this stuff. It's all proteinaceous. It gets in your hair. You've got slime, coral sex, everything. It's really awesome. So, all right, let me keep this down now. So once they start going, everybody's in a tizzy, right? We got to get the sperm and egg. We want to get it back to the boat, get it back to the lab. Well, how do we actually go and collect this snow of coral sex, right? So what we can do is we've actually started to make these tents that we can drape over a coral colony. So then we don't have to actually be next to the coral and wait all night long. We can go out as soon as after sunset, go out, set up these tents, and then go back to the boat and just relax. Because now we know if this coral is going to spawn, all of those gametes are going to be funneled up into this tent, into this plastic collection bottle that we've got. And so then we can come back after the spawning event, collect these sperm and egg bundles, break them apart, cross-fertilize species, and then start developing new coral larvae. Now, after a period of time that these larvae are swimming through the water column, once they start to find a new piece of reef patch of reef that they want to settle on, then they start to develop and grow into an adult colony. Okay, so here's one of those planula larvae. It's got cilia all around it, little hair-like extensions that it uses to swim through the water column. It's got a mouth that forms after a few days, so it can start to eat. Once it starts to settle out, it kind of flattens out a little bit, and you can start to see the start of tentacle buds forming. And then these guys are a little bit further along. Here's a little group of 4 or 5 that have started. And then each of these is going to hopefully grow up into an adult colony. And so once we get all of these kind of growing up, we get back to our full-fledged coral reef. Okay? Now, I mentioned coral reefs are really important habitats, right? So they're the basis of the food ecosystem and the food web in these tropical waters. They're important for food for us as well, right? So we get a lot of seafood from coral reefs. They also provide lots of coastal protection, as well as for those of us that like to go diving, right? It's a huge source of industry and ecotourism. But more and more, we're starting to see coral reefs look like this, right? So as environmental stressors increases, as sea surface temperatures start to increase, as ocean acidification or the pH of our oceans start to decrease, we end up seeing corals that start to look like this. So that we get this paling of the coral color and the coral tissue. And like I said before, corals themselves are basically colorless. And so what we're seeing here is the translucent coral tissue devoid of those algae that are living inside of them. And so we can see the calcium carbonate skeleton that the corals produce and make up for this reef-like structure. And so this phenomenon is known as coral bleaching. And so it's not that anybody's going out with some Clorox and dumping it on a coral and causing it to turn white. It's this partnership, this symbiosis that drives the success of coral reefs is really fragile, and it's sensitive to environmental stressors. And anything that gets out of whack can break open this symbiosis. And the coral actually spits out those algae. It can recognize that the algae are stressed out or they're no longer being a good symbiont. And it gets rid of them in the hope that a better symbiont's going to come around. The problem is these environmental stressors that lead to coral bleaching, increased temperature, increased chemical stress, changing pHs. These are not really short-term events all the time, right? So we can get long-term warming over the summertime, temperatures that well exceed the normal temperature of corals. Once this symbiosis breaks down, if those conditions don't return back to normal, the coral will ultimately die. Because while they have stinging cells just like other cnidarians, there's not a lot of food and nutrients in the tropical oceans. So there's not a lot of food that they can capture. And so they really rely on those algae for their overall success. And so the last couple years, we've had some pretty significant El Nino events that have led to the 3rd massive global bleaching event in recorded history. Okay? And these started around 1998, and we've had 3 massive global bleaching events since 1998. The XL Catlin Seaview went out and monitored this bleaching event that started in 2014 and went through 2016. This is in American Samoa. And this is a reef in December. So remember, American Samoa is in the southern hemisphere, so their summer is our winter. So this is December 2014. This is March 2015. So within a few months, this is the exact same reef from the exact same spot, and almost all of these corals have bleached and or died. And this was what we saw all across areas of the Pacific. The Great Barrier Reef was hugely impacted. You probably saw a lot of the news articles. Some of the popular press even said, they published an obituary of the Great Barrier Reef saying it was dead. It's not dead. A lot of it bleached and a lot of it died, but it's still there. But this is what we're facing now. This is the new reality of coral reefs. These are happening almost every year across the world. Corals are bleaching. And we need to understand how they're going to influence, how they're going to relate to this in the future. How are they going to come and be able to adapt? Or are they going to be adapted? And so that's where kind of my question comes in, is how corals are able to respond to their environment and how their microbial partners, either the algae that live inside of them or the bacteria, are contributing to their resiliency. So to go back and kind of flesh this out a little bit more, right? So corals are the individual polyps. These are the animals. Inside of their cells, these are the unicellular dinoflagellate algae that are producing all of that photosynthesis. And that's the classic two-partner system that everybody's been studying since the 1950s, since we really got onto coral reefs and really started figuring them out. But in the last kind of 20 years, and this goes for a lot of different fields in biology, we're starting to really come to appreciate the roles of other microbes. And by microbes, I mean bacteria, fungi, archaea, viruses, that are all super abundant in these habitats and associated with either the algae, the coral, or both. And so we're really starting to study these guys now because they are members of this partnership. Okay? It's no longer a two-partner interaction. We really have to consider the role of all of these microbes, because the coral can actually structure the community of the bacteria associated with it. In times that are good, it can structure and say, these are the good bacteria that I want. When some of these environmental stressors come about, the coral weakens in its immuno state, and that can lead to a flux in that microbial community and lead to coral disease or lead to a bleaching event. And so the presence of some of these bacteria really might have dramatic implications on the overall health and physiology of the coral. And that's what really kind of keeps me up at night, asking what are these microbes doing? Who are they? What are they doing? And how can we use them in management of coral reefs in the future? And so just an example of how different microbes can alter host physiology. These are a bunch of pictures of different coral diseases. And so these are caused mostly by bacteria that are sometimes normally found in the ocean environment. And under kind of normal conditions, they're completely benign. You might find an otherwise quote-unquote pathogenic bacteria living on a coral, but in times of normalcy, the coral can evade any infection and keep those bacteria at bay. It's when the corals get stressed out through environmental shifts that then these microbes, these bacteria that are pathogenic, can start to mount an attack and have this impact. Okay? So some of these kind of look like bleaching, right? So here's some white spots forming on this colony. This one looks like it's completely bleached. Here's another one. But a lot of these coral diseases are actually tissue necrotic. So the bacteria eat away at the coral tissue. And so it appears bleached, but the coral tissue is actually gone, and all that's left is the skeleton behind it. And so there's about 40 different coral diseases that are out there. And so we've kind of described them, and we know the symptoms. But oftentimes, it's really hard to identify the causative agent. So to find that bacteria that's actually leading to the disease is really tricky, because we can't grow a lot of bacteria from the environment in a laboratory setting. So if I bring a sample back, I can only culture less than 1% of the bacteria found in a milliliter of seawater. The majority of them just don't grow. And so that really limits what I can do in the lab if I'm trying to isolate a pathogenic bacteria that's causing a disease. There are other strategies that we can use to get at that, but I won't go into those tonight. First, I want to get back to the slime. So this is that coral mucus. Okay? This is that excess photosynthate that's produced from the coral in addition to what it really needs for its own energy and metabolism. And so you can see it actually is this kind of sheet of mucus that sits on top of the coral. And it provides a lot of benefit, actually. So it acts as a natural sunscreen for corals. So corals sometimes are in really, really shallow water, and they can be affected by UV radiation. And so by having this extra kind of layer on top of it, it protects it from those UV rays. It acts as a desiccant. So sometimes at low tide, corals can actually be exposed out of the water. And again, by having this kind of viscous layer of jelly sitting on top of it, it keeps it wet, and it doesn't dry out during that low tide. It also provides a foundation of an ecosystem. So microbes are living all in this mucus, because it's basically sugar and protein. Really rich carbon sources for microbes to grow on. And it's what I go out and collect from a coral and bring back, and I study this snot, and I look for bacteria that are associated with it. And it's amazing the diversity that you find in there. And so how do we go collect coral mucus, this slime, and the bacteria that reside in it? Well, we go diving or snorkeling. We go down with just a little syringe, a sterile syringe, and we just gently agitate the surface of the coral. And part of the coral defense, if they get something on them, if they get any sediment or sand, or if a fish comes by and rubs against it, their first response is to slough off that mucus layer. Because if it lands on the coral, the only way to get it off is to get rid of that surface layer. And so as soon as you start kind of messing around with the surface of the coral, it starts secreting more and more of this mucus. And then I just sit there with a syringe and I suck it up. And then I go back to the lab. And so it's a non-invasive approach to get these samples. So I'm not killing the coral, right? The corals themselves tuck themselves into the calcium carbonate skeleton. And I just kind of agitate the surface, and it secretes all of this mucus. Sorry, I keep tripping on myself here. So I take that sample back, and then we can put it on bacterial media in the lab. And then we can identify and isolate individual colonies that are able to grow under those conditions. And so I've got libraries of bacteria that we've isolated from corals that then I can start to screen and figure out which species of bacteria are there. And then I can start to ask questions what they might be doing. Are they helping the coral? Are they potential pathogens? If I change the conditions, what do they do? And how do they alter the host physiology? One really great thing about coral mucus is that all of these bacteria are living in it. All of those bacteria are naturally competing with each other, right? So they're all going in after the different carbon sources that are there. And so one might be producing an antibiotic to kill another species. One might do something else to inhibit motility or locomotion or ability to utilize this carbon source. And so what we can do is kind of use this idea of natural products from these bacteria that naturally live in this environment and harness some of those tools. And so coral mucus is actually the first line of defense against invading bacteria. So this is a pretty simple experiment. We took just natural seawater, okay, just a sample of seawater, and put it on our bacterial agar plates that we can grow in the laboratory. And you can see there's so much bacteria on here that you almost can't even tell individual colonies, right? It's just a slurry and lawn of bacteria. If we take that same exact sample and plate it onto an agar plate, same media, but now we've added a sample of coral mucus to that plate, we get a dramatic reduction in the number of bacteria that are able to grow. And again, this is because some of those bacteria that are living in that mucus are producing chemicals that inhibit the growth or slow down the growth of other bacteria. And so this is kind of natural microbial warfare that happens that we can start to understand and take advantage of. And so there's a lot of different mechanisms that bacteria are going to need to either start causing a disease or even to colonize on a coral host. Surface motility is really important. So if a bacterium lands on one portion of the coral, it has to move until it finds carbon sources that it can use. Okay? So there's two main strategies for bacterial locomotion. One is the cilia. So just like those hair-like extensions that were on the outside of the swimming coral larva, bacteria have little hair-like extensions around those cells too. And they can use those to crawl across the surface. So if they land within this coral slime, they can use those cilia to crawl through until they find a new niche or a habitat that's suitable for them. Bacteria can also swim, so they have flagella, or a little tail at one end. And they can spin that tail and swim through a media matrix. They can also form biofilms. So every morning when you wake up and you feel that kind of gunkiness on your teeth, right? Those are bacterial biofilms that formed as you were sleeping. And that's why we need to brush our teeth, to get rid of that lovely smell in the morning, right? So the same thing happens on corals, is that bacteria grow and they aggregate, and they form this almost multicellular structure living inside this mucus. They have different enzymes that degrade different sugars or molecules as they're utilizing this coral mucus as a food source. Okay, so these are these proteases, enzymes that degrade protein and break down complicated structures. And then pathogenic bacteria, you might imagine, right, if I'm a single pathogenic cell trying to cause a disease on a coral, if I'm one individual bacterium in a giant coral, does it make sense for me to start expressing all of my virulence pathways? Not really, because I'm one in a sea of billions. And that coral is going to be able to mount an attack on a single bacterial cell, right? So that pathogen will start to divide, and more of those pathogens are going to form. And it undergoes a process called quorum sensing. So once a quorum is reached, right, a certain density threshold of all of the same species that are all producing chemicals, they can sense those chemicals, and now I know, all right, there's a lot of us around. And so what they do is they coordinate their expression of some of these virulence properties, and then can mount an attack all as one and start to cause a disease. So here's a look at some of these kind of proteases. So I hope you can kind of appreciate. This is a milk agar plate, so there's skim milk in this agar plate. And this is looking at the bacteria's ability to synthesize some of these proteases that degrade the casein protein in milk. And so you can sort of see, here's the bacteria spotted, and as it's moving through the media, it's degrading that casein, and the agar is becoming more clear. Okay? Here's a look at kind of this quorum sensing. So as the bacteria are growing and increasing in density, they're producing these chemical signals. And once this population threshold gets high enough, they reach kind of a saturation threshold of those chemicals, and they can sense that there's enough of them to start causing disease. And that's when they mount an attack on the host. And then we can also use model systems to kind of figure some of this stuff out. And I'll talk about that in a second. So my big research questions are really kind of looking at how these natural communities of bacteria associated with coral influence the physiology and the health of the holobiont. And by holobiont, I mean the coral, the algae, and the bacteria, all working together as one. So which species of bacteria are present? What are they doing? How do these microbes help the coral? Do they help the coral? And what about invading pathogens that are either normally associated with the ocean environment, or in the case of one pathogen that I study, when we introduced it to the coral reef through mismanaged sewage water in the Florida Keys, right? So we introduced a human pathogen called Serratia marcescens into the coral environment in the Florida Keys. That pathogen found its way onto the corals and found a way to cause a disease. So how do these natural bacteria interact with those pathogens? And can they provide some sort of benefit? And now, I started grad school working on corals. The species that I was working on was ultimately listed under the Endangered Species Act. And so it's now threatened. And it becomes really hard to convince the government to give you a permit to go hack off threatened species from the reef to ultimately infect with bacteria and kill. So midway through my dissertation, I found a need for a different system to work on to answer the same kind of questions. And so now I'm sort of a model system guy. How can we use model systems that nobody cares about, and I can kill as many of them as I need to, in order to answer questions in coral and otherwise help to save them? All right, so I'm going to tell you 3 really short stories because I'm rambling on and I know that we can't stay here all night, even though I would love to. So one is this idea of a model system. And so this sea anemone Aiptasia, it's a small sea anemone. It's in the same class of corals. And I use them now in my lab to study some of these same interactions of looking at the host and the microbial partnership. And so I'll share a few slides about that. Then I've got some data looking at some of these natural associated coral bacteria and their interactions with otherwise pathogenic bacteria that would cause disease on coral. And I'll tell you a little bit about that. And then my work in Stanford really focused on the symbiosis in response to environmental stressors. And so I've started to look at thermal tolerance in sea anemones, and thus corals, using this model system. And so let's start first with the model system. So I've kind of alluded to this, but why is it maybe not always a good idea to study corals when especially if you're a microbiologist and you want to study the bacteria that causes disease and you're killing corals in the process? So there's a lot of reasons that it's hard to study corals in the lab. One, sometimes they're inconvenient sizes, right? So this is a drastic exaggeration, but this is a really large boulder coral that takes thousands of years to get this big, right? And if it takes that long to form, then I don't want to be the one that hacks off a piece of it to bring back into the lab. They're really slow growing, right? So 1,000 years maybe to get this size. The size of a table, 100 or more, right? And so I don't think anybody in science, or especially a graduate student, does not have 100 years to complete their dissertation, right? We need to move a little bit faster than that. Corals are difficult to maintain, right? We know that corals are pretty sensitive to their environment. Anybody have a saltwater tank at home that has corals in it? You're smart people. So they're tricky, right? They're finicky with nutrients, with temperature, with water chemistry. If anything's off, you can start to lose them. And they're really expensive to maintain. Corals also come with this really hard calcareous skeleton, which is essential for coral reefs. But if you want to look at the tissue, that giant skeleton gets in the way. It's hard to put this guy under a microscope, right? And then as I alluded to, for my species, they become protected, which is a really good thing. We should be protecting corals. But if the corals are protected, it's harder to bring them into the lab and mess around with them and manipulate them. So early in or kind of midway through my dissertation, I needed another system. And so John Pringle at Stanford is a model system guy. He's a yeast geneticist by training, and had a long career in yeast genetics, and then kind of at age 60 had a midlife crisis maybe, and decided, okay, I'm done with yeast. I want something else. I don't want to retire yet, but I want to make my mark in somewhere else. And so he wanted to study corals, and he asked around, and he asked what was the best coral that everybody should be working on. And for the reasons that I alluded to in the last slide, there really is no great model system, a coral model organism. So then he started talking to people who maintain saltwater aquaria, and everybody said, well, they've got these little anemones that grow faster than my coral. They come in on the live rock. They're not really attractive, and they're so hard to kill. Well, that right there was reason number one to start using it. Because if they're really hard to kill, they're easy to maintain in the lab. I have only undergrads at Eckerd. I can trust them with these anemones because they can't kill them unless we grind them up. So these are convenient to use in size. They're really small, about 1 centimeter in size at most. So about the same size of a coral polyp. They're soft. They don't secrete a calcium carbonate skeleton. So I can smush these between a microscope slide. I can easily homogenize those tissues. We have large clonal populations. So just like corals that reproduce asexually to form that entire colony, so do these guys, which means my population is genetically homogeneous. They're all the same. We've now done a lot of transcriptomic and genomic sequencing. So we have the genome sequenced for these guys. We've got proteomics being done. We've got transcriptomics being done. So we know a lot about the genes that are expressed under different conditions. Really, really important, these guys are symbiotic with the same kind of algae that corals are. So the color that you see here, this brown color, is from the algae. And then what's really awesome is we can bleach them just like corals do. So we can remove all of the algae from their tissue. And so you see here the whiteness, the lack of color. But what we can't do really with coral is keep corals in this state. It's really hard to keep them symbiotic free because they need those symbionts to grow. But I can simply feed my anemones brine shrimp, little sea monkeys, totally happy. I've kept them this way for years. No worries. And then we can use fluorescence microscopy because the photosynthetic pigment, the chlorophyll molecule, fluoresces red under UV light. And so we can use this as a means to kind of identify some of the strains. All right. For the sake of time, I'm going to skip through some of this stuff. This is just a really short video that I'll just start and then stop. Looking at feeding of our little anemones. So that was a little sea monkey or brine shrimp. It catches in its tentacle. And then because these guys are pretty translucent, right, you can almost see the individual Symbiodinium cells. And then you can track that brine shrimp into the gastrovascular cavity, the stomach basically, where it's going to digest it. All right. So the first story I want to talk about, so quickly running out of time, is Acropora palmata. So this is that threatened species that I talked about in the Keys. It's threatened now because it's dwindled so far compared to what it was in the 1970s that you go out on a reef and it's really hard to find Acropora palmata. This species has been affected by a number of diseases, one of which is caused by the bacterium Serratio marcescens. And this is known as the white pox disease. Okay, so you end up getting these white pox of bacteria that are eating away at that coral tissue. And so here's the bacterium under scan and electron microscopy. And so what I wanted to do is use the anemone system to understand how these bacteria starts to cause disease, and if some of the bacteria associated with healthy corals interact with this bacteria as it's trying to cause a disease. And so I had an undergrad at the time, Nick, who did a lot of this work with me. And what we're using here is the anemone system. This is a false color image, so this is just a black and white image. And what we did is, again, easy manipulation, right? I can put a single anemone in a small little volume of water. Totally happy. Can't do that with a coral. And so what we did is we infected it with different concentrations of our pathogenic bacteria. And as concentration increases, right, this is 10 to the 8 cells per mil. So that's a lot of bacteria, 100 million cells per mil. And what we see is that the health of the anemone starts to decline pretty dramatically. So the tentacles start to shrink in, it contracts. And if we give it enough time, it actually turns into kind of a soup. So the tissue sloughs off and the bacteria eats away at what was an anemone. And so it's pretty easy to tell when an anemone dies because it just turns into this really stinky ooze. More slime. It's really kind of fun. We can quantify this using Kaplan-Meier survivalship curves. And so at the higher concentration of our bacteria, of our pathogen, the survivability decreases. So we get an increase in mortality with the higher concentration of cells. And this was important just to show that these pathogens are actually able to cause the same phenomenon that we see in corals in our sea anemone model. So then what we wanted to know is how do these bacteria that are normally associated with corals interact with these pathogens? So one thing that happens is when you put this pathogenic bacteria onto coral, it starts to increase its metabolism. So it really likes all the sugars, and it grows way faster than it does in normal laboratory media. So when grown on mucus, we see this growth is way higher. And so it's specifically utilizing the substrates, the carbon sources in that coral mucus. We have a little reporter. So we have now a blue pathogen if it's able to utilize a certain substrate in my media. Okay? So if it can grow on that substrate, it turns blue. Right? And this is just a positive E. coli control. Then what I did is I took some of our coral isolated bacteria that we got from healthy coral. And instead of this black line down the middle, I streaked the bacteria. So this is a coral bacterium from healthy coral. And then I spotted the pathogen or my positive control E. coli right next to it. And what we see is we get inhibition of that blue color. Right? So now there's interference from the coral bacteria on the front of the pathogen that's next to the bacteria here, we don't see that blue color. On the other side of the colony, we start to see blue. So there's something produced or some sort of interaction going on between the coral bacteria and the pathogen that is limiting the ability to grow on this media. We also then wanted to see, do these coral bacteria affect motility of our pathogen? So I mentioned swimming and swarming before. So swimming is due to that flagella, that little tail-like appendage, and they can swim through a medium matrix. And it's hard to see, but there's kind of a halo. So the bacteria were spotted in the middle, and there's a little halo coming off. Swarming is based on those cilia, those hair-like extensions, and it crawls across the surface. And this pathogen displays a really quite beautiful spreading out across the colony in this really kind of fine dendritic pattern. We then mutated our pathogen to not be able to move at all. So if you put it in a swimming media, it just stays put. If you put it on a swarming media, it can't move any further. Okay? And then we asked, well, is motility important for the ability to start causing a disease and kill our host? And indeed, it looks like it does. So even at the highest concentration, we have a greater survivability in our sea anemone than we did. We have 100% mortality under the wild type condition. But now if we knock out that ability for the bacteria to move around, we don't get progression of that same disease. And so we're starting to kind of check off some of these virulence properties in these bugs. Then we wanted to see, all right, so if we knock it out genetically, right, we create a mutation and it can't swim, it lowers the infectability of the pathogen. But how about if we interact that pathogen with our coral commensal bacteria? So here's just the pathogen alone next to just an empty filter disc with no bacteria on it. And we see this surface spreading, right? That's swarming. Then I took some of those same coral bacteria that inhibited the metabolism of the pathogen and then spotted them next to the pathogen. And what we see for 5 out of 6 of these coral bacteria that are on this filter disc, we see this kind of zone of inhibition of swarming right around the colony. And so it's not killing the bacteria, but it's stopping the ability of that bacteria to move around. We have a question?
We've got a question from Chris, who is tuned in with us on Facebook Live. Chris wants to know, is whitepox caused by pathogens commonly found in sewage?
So the question was, is whitepox commonly found associated with water, associated with sewage? Yes, in this case, it was. So the way that Serratia marcescens was entered into the coral reef environment, some source tracking studies have pinned it back to mismanagement of sewage in the Florida Keys. So that raw sewage was dumped into the ocean environment, and that's the source of it. And so now it's associated with these reef environments, and given the right opportunity, can start to cause a disease. Thank you for the question. So now we see there's some coral bacteria that are able to limit the metabolism of this pathogen and also slow down its motility. And we know that motility played an important role in the infectability and the mortality of that causing disease. So then what we did is a really cool study where we took our anemones and we either infected them with our pathogen, or before we infected with the pathogen, we took these good bacteria and put them on the anemone first. And then we put the pathogen on and looked to see what was the result. And so on the top panel here, this is just the pathogen alone. And as we saw in the first couple of slides, as time progressed, right, the anemone disintegrates away and dies. So the pathogen is able to cause that disease and kill the anemone. What we see when we inoculate first these good bacteria and then subsequently infect with the pathogen, we see a little bit of a reduction. So maybe they got a little bit sick. But then by the end of the experiment, and this is the same anemone over 7 days, it looks completely healthy. So we were able to slow down or even stop disease progression with this pathogen by pre-inoculating this anemone with those quote-unquote beneficial bacteria. And so from this study, what we see is that these enzymes that are utilized by the pathogen to degrade some of this coral mucus substrate are induced when the pathogen sees coral mucus. So less food are available, it's got the enzymes needed to grow, and it grows really fast. We see that some of these bacteria interact and inhibit some of the virulence properties necessary for that pathogen to start causing the disease. And then mixed together, they can really dramatically alter the host. So the pathogen alone can kill the host, but you put these coral-associated bacteria on before a pathogen, and we might actually slow down disease progression. All right, I'm going to have to skip thermal tolerance. I'm sorry, but I'm going to kind of skip ahead. Sorry, Brian's going to kill me. And just tell you one slide about what we're doing now at Eckerd, and then I'll open it up for some questions. We can chat a little bit more. So Eckerd College is a small liberal arts college down here in St. Pete. We have a really great marine science and biology program. I'm lucky to be a member of both of these disciplines. And we're using Aiptasia as our model system to study corals. We hope to get out in the field. We're actually doing some coral restoration work this summer down in the Florida Keys. But during the semester, this is how I keep my anemones. These are literally food quality Tupperwares. So when you go to Chipotle and get your burrito, right, these are the plastic versions of what your salsa is coming out of. And the anemones don't care at all, right? They don't need flow through. They don't need special seawater conditions. I get seawater from the bay, plop them in. They're totally fine. Maybe change the water once every week or so. Can't do this with corals. So this is really easy. They're on a shelf with a light. That's all we need to set up. And so some of the projects that we're getting into in the lab now is looking at the overall community. So I mentioned before, one of the questions, right, is who's there and what are they doing? And so the first thing we need to do is both as a culture-based, so trying to isolate bacteria from the anemones, but also I mentioned we can only culture a very small subset, right? So we're taking DNA-based approaches. We're isolating all of the bacterial DNA associated with these hosts and sequencing all of the DNA, and then trying to put pieces back together and identify which bacteria are present. And then once we know who's there, then we can start to ask questions, maybe what are they doing, right? We can do those kind of interactive studies that I showed you with the anemones to piece together, is this a good bacterium, a bad bacterium, what is it bringing to the table? And so that's getting in those host-microbe interactions. We're also doing microbe-microbe interactions, okay? So those that are isolated from healthy individuals, how are they interacting with pathogens? And then we're getting into this really awesome project that I'm calling germ-free or bacteria-free Aiptasia. And so I mentioned we can eliminate all of the algae associated with these anemones, right? But now I'm trying to remove all of the bacteria that are normally there. So that I have a completely sterile anemone, which then I can go back to that study where we added those good bacteria, right? When we added those good bacteria, all of the normal bacteria associated with that anemone were already there, right? So we added 6 new members. But what happens if we have a blank canvas of an anemone, and now we can preferentially add back 1 strain, 2 strain, 100 strains, and make our own microbial community? Can we kind of direct what that community looks like and what kind of impact it'll have on the host? And again, because we can grow them in really small dishes, right? So this is a 24-well plate. We can put the anemones in, and what we're doing is right now testing a series of antibiotics that's going to drastically reduce or remove all of the bacteria associated with the anemones. And then we can propagate them under sterile conditions. And then we can start adding back individual strains and ask really what those bacteria are doing and how many different types of bacteria do these anemones need to be fully functional, right? Is there a core kind of microbiome, right? That's a big buzzword in science today. Is there a core microbiome that governs the interactions, or can they get along with just a few? And how does that look? So if you're interested in some of this work and you want to follow us, I'm on social media. I'm not an avid tweeter, but I'm trying to get better. So you can follow me here. My website is here. You can also find me through Eckerd College. So in my lab at Eckerd, we're doing cnidarian physiology. And we're looking at how the coral host, the sea anemone host, and this interaction with its microbial partners influence the overall health so that we can stay on this side of the picture healthy reefs, and not be on the other side. Thank you so much for coming. It was a real pleasure to be here. Thanks. So I'll happy to take any questions you have. Yeah?
Okay, I'm coming to this with no background knowledge of coral whatsoever. So my question is, well, I have several. But
Great.
One of them is, are Aiptasia, those are not corals?
They are not corals.
Okay.
They are sea anemones, so they're kind of cousins.
They're cousins?
Yes.
Okay. What is their natural lifespan?
That's a great question. So there's been very little study of the natural history of Aiptasia, of these little sea anemones. Mostly they are known in the kind of aquarist realm of those keeping saltwater tanks, and they just want to kill them. So there's actually a chemical called Aiptasia X. It's a little cementing agent that you inject into the body of the sea anemone. It hardens and kills the anemone. The problem is they grow faster than anything. And so you can't see the really little ones. And so as soon as you kill an adult, the little ones start to grow up. But it's a big problem in the aquarist trade. So nobody really studies them in their natural habitat. Aiptasia are found on mangrove roots, on dock pilings. I have kept Aiptasia. So these populations of anemones that we've got all originated from a single anemone because they reproduce asexually, and they're all clonal. So in 2003, we started with one anemone. Now we've got thousands and thousands. I have kept a single anemone myself alive for 7 years, but really they have no real lifespan. They're nearly a ball of stem cells that continue to regenerate. And so if they're not reproducing fast enough for you, just naturally through asexual reproduction, I can also go in and cut the anemones. Because of their radial symmetry, I can cut them in almost any way possible and get 2 anemones, 4 anemones. The most anemones I've gotten out of one is 16 anemones that all functional within a few days. You can't even tell that I cut it. So they're basically a ball of stem cells, and they won't ever die unless you kill them, which is awesome. Any other questions? Yes, please.
How do you see like artificial reefs helping or not helping your research in terms of just anything really?
Yeah, that's a great question. So sorry for Facebook. I'm supposed to repeat the questions. So the question was, how do I see artificial reefs either benefiting or influencing, interacting with my research? One, it's a critical part of, I think, restoration efforts and rehabilitating coral reefs to some semblance of maybe what they were. And so the idea of artificial reefs is putting down sort of artificial substrate that then coral larvae can attach to, right? So they have to have some suitable substrate as they're swimming around to start forming a new colony. And so the idea is to put new habitat either in areas where the habitat wasn't, or if the old habitat has been degraded, add back some sort of substrate that those larvae can land on. And I think it is a really important tool. It's still sort of in its infancy. And so the projects that have been going on haven't had a lot of long-term analysis to see the kind of the success. But there's one organization in specific called Reefball that puts down kind of these calcium carbonate cement structures that then you can either sort of glue adult colonies from one area and kind of start seeding that new substrate, or you can have recruitment of juvenile corals into that. And I think it's a really important aspect of our research, because if we're trying to understand, one, how corals are declining, we also need to think about how we can better facilitate the success of the corals that are there. And so if we can take corals that are successful in a certain environment and understand what about them makes them successful there, then maybe we can use some of that to guide or specifically use some species or genotypes of coral, plant them out on these artificial substrates, and then start to get new reef formation. So I'm starting some projects looking at coral reef restoration down in the Florida Keys. And so here we've got corals that we grow in either laboratory settings or in kind of protected areas offshore. And we study those individual corals for their resiliency, for how well they're going to respond to the environment that we put them in. And so in future management, it really becomes important to understand something about that species of coral so that you know if I take this individual and put it at this reef site under these abiotic conditions, it's going to be successful there, right? Because we don't want to put a lot of resources and effort into outplanting corals to make these artificial reefs, only to have, you know, really weak corals or those that are sensitive and susceptible to disease or bleaching, if we know that that's an environment that is hypervariable. And so it becomes, with artificial reefs, one, having the substrate available, but then two, the other half of that is to understand the biology of that coral species individually, to know where should we place these corals to give them the greatest chance for success. Facebook question.
We've got another question from Chris, who's joining us on Facebook. Chris wants to know, is pre-inoculation practical outside the lab? And what is the best approach to dealing with coral diseases in the real world?
Yeah, that's a great question, Chris. So that question was, is pre-inoculation of these quote-unquote good bacteria or probiotic bacteria feasible or practical outside of the laboratory? And what was the other half of that? And how do we use that to manage coral diseases in the real world? So this is like the $1 million question for my research, right? So if we can identify this community of good bacteria, right? Nobody is going to give me a permit to make this cocktail of bacteria and go spread it out on the reef, right? Huge amount of unintended consequences that are probably going to happen there, right? These bacteria may be good for one species of coral, but may take over the rest of the world if we just release them into the environment. But getting back to this idea of artificial reefs and restoration efforts, right? Those corals that we're growing in a laboratory setting and then ultimately transplant them back out into the reef environment, if we can manipulate the microbial community of those corals in an artificial setting and get those bacteria to actually stick so that when we put them back out into the reef, now they kind of have this, they've got their probiotics and they're healthy and they're resilient. And if they maintain those bacteria, maybe they'd be better resistant to infection in the future. And so that's kind of a long-term application that I envision, that as part of restoration efforts, if we can understand what role these bacteria are playing, we might be able to manipulate them in a laboratory setting, get them established, and then transplant them back out into the reef environment where they might have a better chance to survive.
Do you think that climate change is potentially going to affect the timing of going back to the beginning where you're showing the video where the eggs and the sperm are being released? Is it potential that could affect that? I know, the plant world, we're worried about blooming times and migration patterns, and is it a concern that you're looking at?
I'm not doing a lot of actual spawning work these days, but it is a concern.
Can you repeat the question?
Sorry, yes. So the question is, with warming sea temperatures, is that going to influence the timing of coral reproduction in these mass spawning events? Is it going to either shift it so that the corals get confused, or probably going to shift it so we get confused and miss the spawn? But what kind of are the concerns or worries there? Absolutely, it has implications. It may shift the cycle. So a lot of the cycle that leads to the spawning are based on the lunar cycle and the tides and gravitational pull. Beyond that, we don't really know what temperature really plays into it. Probably more likely is that the temperature is going to stress out the corals, and then they may not be providing enough energy towards reproduction. And so the overall amount of spawn that they're making is going to be decreased. And if we've got limited spawn, then the chances of those sperm and egg actually cross-fertilizing, leading to those planula larvae that then are going to settle out and recruit, are going to decrease. So it may drive down ultimate recruitment because those colonies that are supposed to be producing those sperm and egg are now worried for their own survival and are not going to be devoting a lot of energy into gamete production and reproduction. Yeah. Another question?
Once coral bleaching happens, is that skeleton, can that skeleton be used by new coral animals, coral larvae?
It can, absolutely. So if the coral that was there, if the tissue die, and all that's left is the rocky skeleton, that is barren substrate that is prime real estate for new things to grow on. And so corals are one that we would hope that would land on it. The problem in the reef environment is So some of these corals only reproduce once a year. And so that spawn that I showed you, that happens in the full moon in August once a year, and that's it. So if that coral dies a couple weeks before and now there's open substrate, then maybe those larvae have a chance to get on there. With that prime real estate, everybody else is going to compete for that substrate too. And the problem is corals are the slowest growers kind of in the coral reef environment. So algae is going to colonize that barren substrate much faster, and it's going to outcompete the coral for that space. So yes, corals can queue into substrate that was left behind from the same species, but oftentimes that openness of that substrate is not going to last long enough for a new coral larva to get in. Or even if it does land, again, that one individual polyp growing is going to take years. Algae can do that in a couple days. And so the juvenile corals are at a real disadvantage competitively for that space. Anything else? Yeah.
Is most of your research or the general field concentrated on hard corals, or is it also including soft corals as well?
Great question. So the question was, is the research that I'm doing and in the field in general focused on hard corals, or are people studying soft corals as well? I would say there's probably a predominant bias towards the stony corals, those that are actually secreting these big reef-like structures. Especially now, a lot of people are focused on ocean acidification and the effects of decreasing pH and the effect that that has on the calcium carbonate skeleton. So calcium carbonate is stable under slightly basic pH, which the average pH of the ocean is around pH 8, so just slightly basic. But as more and more carbon dioxide is being dissolved into the oceans, it's lowering the pH, becoming more acidic. That calcium carbonate dissolves under lowered pH conditions. And so we're starting to lose the reef that is present because of this lowered pH, as well as corals have a harder time making new calcium carbonate under this acidified conditions. So from that standpoint, a lot of energy and focus is going on to the stony corals, those that are producing those reefs. Soft corals, we call them soft, but they're still making calcium carbonate, just not to the extent that the stony corals do. And so people are not studying them in terms of kind of ocean acidification from the calcium carbonate standpoint, but they're interesting nonetheless because they're also affected by bacteria. They can be diseased. They also have Symbiodinium, so they're symbiotic with the algae, so they can undergo bleaching. And so there are some things that soft corals, without having a lot of that calcium carbonate, is sort of sometimes naturally easier to study for some of those questions. But the majority of people, I think, are researching shallow water stony corals. But as our stony corals are declining, our soft corals are increasing in abundance. So if you go diving in the Keys now, you're going to see way more Gorgonian sea fans and soft corals than you would some of the stony corals. So we're kind of getting a shift in the abundance of these species, which may drive an increased interest in those soft corals that are out there. Yep.
Got a question here from Bren on Facebook Live. Bren wants to know, do the anemones you work with bleach the same way as corals?
Great. So Bren is asking on Facebook, do the anemones bleach in the same way that corals do? They do, absolutely. So they're a little bit hardier than corals, so it takes a little bit of a higher temperature to get that breakdown of the symbiosis. But if we increase the temperature up to a threshold, the anemones will spit out the algae. And this is what I didn't have time to really talk about tonight, but we can quantify that bleaching response. Because of the fluorescence of that chlorophyll from the algae, we can take those anemones, grind them up, and pass that homogenate through a machine that has a little laser. And as the cells are passing by that laser, if that fluorescence is there, the machine detects it. And we can get really accurate and precise counts of how many algae are in the anemone remaining. So we start at day 0 before any heat stress, and we call that 100% populated. And then we expose the anemones to heat stress over a period of time. And then we subsample as that stress is going on, and we can count literally almost every algal cell that's in the anemone and get a really finite look at that bleaching response. But yeah, they do lose algae in the exact same way.
Okay.
Yeah.
One more on Facebook, and well, let's keep going as long as let's
follow up with that question about
Yep.
the symbionts being expelled either by the anemones or by the reefs.
Mm-hmm.
Is it because they're detecting that those dinoflagellates aren't photosynthesizing effectively, and they're just saying, we're going to get rid of you, and I'm hoping to pick up a better version?
Yeah, so the question is, so what's kind of the mechanism of bleaching? How does the coral know that it wants to expel or should expel some of these algae? And so we know mechanistically that it is expulsion. That is the primary mechanism. The why is the big question, or how the corals know. So we don't know if it's the algae that are signaling to the coral that things aren't good, if the coral recognizes that they're no longer photosynthesizing. So we've got data that we know that under heat stress, the photosynthetic efficiency of the algal cells diminishes, so they're not producing as much photosynthate. And so that might be a trigger for the coral. I'm not getting as much food as I was, so you're out. The thermal stress also affects the algae. And so they might be producing toxic chemicals as those cells are starting to die that become toxic to the coral host. And so once it recognizes those, it might say, this isn't good, let's get out of here. And then the idea of kind of bringing in a different symbiont is known as this adaptive bleaching hypothesis. So if one symbiont is not functioning well, get rid of it in the hope that you might reacquire another type of Symbiodinium or a different strain of algae that is more able to deal with that stress. And so within the realm of these dinoflagellate algae, there are some that are more resistant to thermal stress than others. Some of that comes with a trade-off sometimes. So those that are more thermally resilient maybe inherently don't produce as much photosynthate. So they're not as good of a symbiont in terms of what they're bringing to the table, but in times of stress, they're more resilient. And so there's a lot of work going on with this idea of, yeah, if one coral bleaches and it has a variety of Symbiodinium to choose from, right, which ones start to populate again? And so there's a lot of differential bleaching that we've done in the sea anemone model too, because one thing that I didn't mention with the sea anemones that we can do is if we make them symbiont free, right, we remove all the algae, I can then take Symbiodinium from a coral, put it in the anemones, and then test how that Symbiodinium does in that anemone background. So I can take the same genetically homogeneous population of sea anemones, infect them with different strains of Symbiodinium, and then heat stress them and see who does better. And so we can start to ask the question, is it the host that is bringing stuff to the table in terms of thermal tolerance? Is it the algae? And now I'm starting to get into, is it the bacteria? Because everybody's been ignoring the bacteria for so long. Ignoring is a bad word. We haven't ignored them. It's complicated to study them because there are so many of them, and we can't grow them all in the lab. And so to really tease apart what's going on becomes really challenging. It's a lot easier if it's one species of algae, one species of coral. It becomes harder when you throw in, 500 species of bacteria, 1,000 species of bacteria. So I don't know what I'm getting in for, but we'll see what happens. Do we have one more on Facebook?
I have heard two views about reef-building corals. One, reef corals are one single individual made up of many polyps. And two, reef corals are many individual but identical polyps that are connected together. You hinted at this, but how do you see it?
Great. So the question is a coral colony a single individual with multiple polyps, or multiple polyps making up a single individual? You're right.
The second part was
it's tricky.
Reef corals are many individual but identical polyps that are connected together.
Okay. Yes. So a coral is a group of many polyps that are all physically connected, so they all share a digestive system. So when you look at a coral colony, right, a big boulder, those are many individual polyps that are all connected and have been derived from a single coral larvae that settled out and started dividing asexually. Okay? So genetically, across an entire colony, theoretically, those are all genetically identical polyps. They are all connected at the base within the calcium carbonate skeleton so that there is food transfer from one polyp to another. Symbiodinium can transfer from one polyp to another. So it's a single animal made up of hundreds, if not thousands, of polyps, all derived from a single larva that settled out. And so you can think of my little sea anemones as kind of one of those polyps. But the sea anemones do not form a colony. They're always solitary. Okay? Which makes my life a lot easier, because then I've got one individual and one polyp, rather than one individual and 1,000 polyps. All right. I think I've kept you here long enough. Thank you all for coming. Facebook, thanks for joining us. It was a real pleasure to be with you. Thank you.